let's see. So our next group is from MBX Biosciences, which you guys have been hearing about. Another company that we're really excited to partner with. They had us come out to their be part of their meeting last week and be able to talk to all of their people. So they welcomed us with open arms as well and are willing to collaborate with us in their trials. So please welcome Dr. Sam Azoulay, who's the Chief Medical Officer at MBX Biosciences.
Thank you. Thank you. Good afternoon. Just let me start by saying I'm sorry that the product doesn't work for alopecia. I would have loved to, but so first I would like to start by thank you very much, Patty and Michelle, for inviting MBX to this meeting. I think I've been in the business, in the pharma business for long enough for having interacted with a lot of patient association. I've always been impressed by the quality of the interaction, how focused the audience is. I mean, think about it. We are Sunday afternoon, 3 p.m. almost, and we are here. And also the curiosity you have with the science. That's very, very impressive. I mean, obviously it's about you, right? So that's why there is so much interest. And in the pharma, as a physician, I don't see patient anymore, and it seems to me that I'm working for you. So that's what we are here for, delivering new drugs that will make you feel better, and that's really the goal we have. So having said that, I'm going to start with giving you a really short overview of the company, and then on our platform, and then before moving to MBX 2109, which is our lead product, okay? So I think that should work, yeah. So the company was created a few years back with really global leader in peptide drug design and development. In fact, with the University of Indiana and one of the most famous person in peptide design, which is Richard DeMarchi. What we call proprietary precision endocrine peptide, it's really a platform which is designed to have optimized pharmaceutical properties, and really with a goal to extend the time action of the product, consistent drug concentration, and we will see why we are aiming to that by design. And at the end, to have, for you, less frequent dosing than unmodified and modified peptide therapies. So we have a few projects already programmed ongoing. In fact, we have two rare endocrine design programs, in clinical development. The first one is the MBX 2109 for hypoparathyroidism, for which we got the orphan drug designation in the US. And the other one is the MBX 1416 for post-bariatric hypoglycemia, which is another rare disease which happens following surgery, gastric surgery, when people are obese and we want them to lose a lot of weight, in fact, and they have this hypoglycemia following surgery, which is really, really life-damaging. And in fact, I'm sure you have heard about GLP-1 agonist, right? Everybody has heard about GLP-1 agonist for losing weight. Here, it's an antagonist. That's the opposite action. And we have also in development obesity, program in obesity with MBX 4291, which is pre-IND, that means that we are completing the tox studies to get the product into human, and that should happen next year. In fact, we have also some other candidates for obesity in this field. And we were very, very happy, in fact, that in September we became public. We became a public company in September of this year. So I'm going to tell you a little bit more about the PEP, right? And what do we want to do and how we are going to do it. So first, we start with advanced chemical modification. And the goal is to enhance the physical properties, including stability and solubility for the product, for the molecule. Increase potency, that means a more potent drug, with multiple potential mechanisms of action in a single peptide. So that means with a single peptide, you can aim to target multiple receptors with a different action. And especially in obesity is extremely important. The second pillar, if you want, of this technology platform is a programmable drug to precisely time the conversion of the drug from a prodrug to an active drug. And really the aim, again, is to reduce the peak-to-trough ratio. And just what does it mean, peak-to-trough ratio? When the peak of the drug, the concentration of the drug in your blood, right, that's the peak, and the trough, that's the minimum of the concentration before the next injection, right? And you want to reduce these variations, this ratio, in order to maintain the concentration as stable as possible, as long as possible, right, to avoid the fluctuation. And we will see it. And there is another pillar, which is a fatty acylation. Fatty acylation that we put some fatty acid on the drug, if you want, to extend the life of the half-life of the product. And again, you heard yesterday about the half-life of the product. What it means that a half-life means 50% of the product is eliminated at what time it's eliminated. That's the half-life of the product. Longer the half-life is, longer the product stays in your blood, right? So that's the goal. That's theory here. That's a design that we wanted to. Now we'll see how we make it happen. We made it happen. So here, on the left side, you have the existing peptide drugs. And you see this fluctuation, right? You see this fluctuation can be over time, can be within an hour, can be within a day, can be. That's what we want. And that usually it's a short half-life, and we see this fluctuation in, again, peak to trough ratio, right? You see the peak and the trough, and boom, and every time you inject the drug or you give the product, it goes up and down. And that usually goes with frequent dosing. So now we look at what we are aiming for, what we call this PEP technology, is to extend the half-life, to deliver a constant drug concentration, and also have less frequent dosing. That's the goal, right? That's now I'm moving to what is of interest for you, which is MBX 2109. How did we make it work? So this drug has the potential to be a once-a-week product. So I'm not going to go through the cause, right? You know it. The symptoms. Obviously, you know better than I do, so I'm not going to go through them. And I go directly to managing the hypoparathyroidism. The standard of care, again, you know it, calcium, vitamin D. And today, Yorvipath, which personally as a physician, again, think is great progress when I'm hearing from patients that their life has changed. Well, success. Well done. I think I'm happy, really happy. It makes me happy. So there is another one, and you heard about this, eneboparatide, and there is another one, which is the oral form. And now we have our product also with a weekly. And that's what we, on the right, that's what we are aiming for, to have a lower peak-to-trough ratio, normal serum calcium, reduce the urinary calcium excretion, eliminate the pill burden, and the convenient weekly dosing, they should ameliorate, improve the quality of life, and reduce the complications, long-term complication. Okay, so that's what the potential. So now I'm going to ask you to stay with me for a few minutes in terms of mechanism of action. How does it work? Remember, two things, prodrug and fatty acylation, right? So in red, the helix you see on the red, that's the inactive drug. It's inactive, and you see the two yellow pieces at each extremity. That's the fatty acylation, right? So when you give the drug to a patient, we inject the sub-Q, so when you it's inactive by design. It's a prodrug, and it's a prodrug, so it doesn't work. It's not active. You inject the product with these two fatty acylation. This fatty acylation, this fatty acid, help linking to albumin, to bind to albumin, right? So then, because of the pH, physiological condition, under physiological conditions of pH and temperature, right, you have one of the extremity, the N-terminal, you see the yellow piece, breaks, and there is a cyclization of the red helix, which is now becoming a green helix. Now the product has been transformed in a drug, active drug, right? Following me? So now this green helix can bind to the PTH receptor. But the beauty of it is that there is still a fatty acid that it stays, right? At the end of the green helix, you see the fatty acid, right? This one? Yeah? This one allows still to bind to albumin. So albumin is also being used as a reservoir, and that slowly releases the active drugs that will bind to the PTH receptor. So that's the technology. This fatty acid is relatively new, but in fact has been already used with semaglutide for obesity and diabetes. So that's how they moved from once a day to once a week. So it's not a brand new technology. It's already, and we know that it's working, okay? So we've performed a phase 1 trial, what we call a SAD, single ascending dose. So that means you progress, you go slowly, you give a first dose and the second dose, and you increase the dose each time, and a multiple ascending dose in healthy subjects, not in patients, in healthy subjects. That's a way of doing a phase 1 study. We start by subjects who are completely healthy before going to patients. The goal of the objective of this study was really to evaluate the safety and the tolerability. And then, as a secondary objective, that's the pharmacokinetic. That means how your drug will behave in your blood, right? How it's going to, you're going to measure the concentration. And the pharmacodynamic, that's serum calcium. That's the serum calcium, and you are going to measure it. When you give serum calcium to healthy volunteers, remember this subject have PTH, normal PTH. What would you expect to see on the PTH? Going down. Going down. So what you would expect, right? PK, as I promised you, right? So I'm saying it's a weekly administration. That's the things that you're going to expect to see. You're going to expect to see an increase in calcium, serum calcium, and a decrease in PTH. That's theory. That's my theory. That's what the assumption you're making before giving the drug. And obviously, you want to be safe and well tolerated. That's a done deal. So here, in terms of safety profile, tolerability, we had two major. The main adverse events were injection site erythema, which is in fact were 25% and was not dose dependent, and disappeared without any medical intervention, very mild, and this was not a problem. And we had COVID, remember? You remember COVID? And we had what you would expect, again, some cases of hypercalcemia at 600 and 900 microgram, at the highest dose, if you want. At the highest dose, we had some cases of hypercalcemia, again, mild and resolved without any medical intervention. Okay, so that here there is no off-target event. There is no treatment-related, serious treatment-related adverse event. And I already spoke about the mild reaction and the hypercalcemia. So now, is it the PK I want to see? yes, it's exactly the PK I want to see. And what we call it flat infusion-like profile. And remember what Dr. Kahn's presentation, where she said that Dr. Weiner made an experiment or a trial where they infused PTH and they observed exactly what they wanted. So that's why it's our goal, to infuse a drug to have an infusion-like, to give the drug and have an infusion-like PK profile. And that's what you see here. So the bump you see each time, it's following 4 injections. So at D0, each time you see 0, that's the day of the injection. Yeah? It's a total of 4 injections. A total of 4 doses which were tested. 200, 400, 600, and 900 microgram. Okay, so that's what, and you see the 900 being the highest. And you see that there is a slight accumulation of products. That's the reason why it increased. And the steady state, what we call the steady state, it's when you get the concentration steady, right? That's a steady state, is reached around 2.5 days, between 2 and 3 days. So the good thing is that the half-life is between 7.7 and 8.9 days. That confirms that this drug can be given weekly. That's what we were expecting. That's what we are seeing. You remember also that I said we want to see a good ratio, or the low peak to trough ratio. It was 1.49 to 1.79, which is, again, after a week. The variability between subjects is low to moderate, and I think we see also a good dose proportionality. That means that more drug you give, more concentration you get. Okay, so you have this flat infusion-like profile staying after a week and confirming that you can give the drug for a week, and with low peak to trough ratio. That was, and the Tmax, maximum concentration, at 2.5 days. That's exactly what, now the reality is confirming the theory. Calcium, that's what you expect to see, some increase in calcium. So here, remember, 4 injections, right? And you see that more drug you give, on the left side, the placebo, 200, 400, 600, and 900 microgram. More drug you give, more calcium increase you see. And at 900 microgram, you see the maximum. Here, that's the maximum change of serum calcium over time, okay, across dose. And the maximum seen here, but there is a standard deviation, that means the variability, is 1.1 milligram per deciliter. So that's a change. Remember what we said also, we see also at 400, 600, and 900, there is an effect. At 200, there is no effect. It's placebo-like, if you want. Remember, PTH, you would expect to see a decrease, right? Look at the placebo on the left, and you see a decrease in PTH. So it's exactly what you would expect to see. So nice results in terms of pharmacodynamic effect, showing an increase in calcium, decrease in PTH, and you see that the decrease in PTH is maximum at with 900 microgram after the third injection. Okay? So here is a summary of the PK. I don't need to get through again the summary. I think we are aligned with the picture of the product, right? So now we are in phase 2. So we move from phase 1 now to the patients. That's the logic. Yesterday, remember, when we went through phase 1, phase 2, phase 3, etc. So here, then we are going to the phase 2. So the phase 2, there is multiple parts in this phase 2. The first one is the optimization period, during which we want the patient to be at the right level of serum calcium with active vitamin D and calcium supplements. That's before randomization. When we have reached this stage, the patient is randomized between 4 arms, between 4 groups, if you want. Placebo, right? The 400 microgram, the 600 microgram, or the 800 microgram for 4 weeks. 4 weeks. And during these 4 weeks, we do not change the dose. After 4 weeks, the patient gets into the second period, still placebo control, during which we can titrate up to the right dose with the objective of keeping the patient within the normal value of serum calcium, but with independence, no active vitamin D at all, and calcium supplement being at 600 or lower than 600 milligram per day. That's the goal. And if this patient responds to this, what we call composite criteria, with these 3 values, these 3 parameters, well, we call the patient a responder. And at the end, after the 12 week, we calculate how many responder in the placebo, how many responder in the treatment arm, combine, you pool them, right? And you make a comparison, and you would expect, for now, I haven't missed my promises, but this one I still don't know. Maybe next year we'll have the chance to show you the results. That's what you would expect to see a difference with placebo. At the end, the patient will have the opportunity, the option to get into the open label extension study for 2 years, where the placebo will be replaced by active treatment. That means that all patients will be treated this time. There will be no more placebo. All the patients will be treated. We'll be looking at urine calcium. You heard how important it is. We'll be looking at vitamin D. We'll be looking at bone biomarkers, tolerability, safety, etc. And we'll also what we call population pharmacokinetics, which is looking at the PK into patients to see if it's similar to what we have observed in healthy volunteers. And we will also be looking at quality of life, patient-reported outcome, PRO. That's a quality of life. So now I'm going to show you not the results of the US election, because I don't know them. We will have to wait from, I don't know when. But here is a site, the representation of the site in the US. We have also a few sites ex-US. So that's, and really enjoyed the panel discussion with the patients. Participation to clinical trial is so important, participating to clinical trials for you, for us, for everybody. It's really because without patient in clinical trials, there is no data, right? And when I heard, I think, someone saying that she was on placebo, and she understood why she was on placebo, even she would have preferred to be on active, which I understand. But that's really the beauty, and thank you very, very much for participating to clinical trial. Without you, there will be no studies, there will be no results, and there will be no product. So, yeah, that's... So I hope that next year, my hope is next year, I will have nice results to present and we'll be on stage talking about your experience with this product. So that I'm aiming for. So really, I would like to thank you for your attention, and I'm happy to answer questions.
Who has questions? Over there, Bob.
My question is will there be assistance to patients to the sites?
It's, do you mean when the product will be marketed?
Yes.
Yeah, it's way too early. I imagine, I cannot say yes or no, but we are...
I mean the clinical trials.
During the, sorry?
The clinical trials.
She means, will there be, is there assistance for patients to travel to the trials?
Yes.
Oh,
Travel
yeah. Absolutely, yes. Yes, so we try to facilitate the life of the patient as much as possible. And I would like also to take the opportunity to thank you, because the FDA is requesting it, but when I was at Pfizer, we started at that time, we involved now patient into the protocol writing, because we want also to be realistic and not overwhelm you with a lot of exam, a lot of visits, which are not necessarily needed. So yes, answering your question, yes, we will be supporting. And I'm taking the opportunity to say also that we have 10% of the company here. We are a small company, so that's why it's 10%. And they are leading, two of them are leading the clinical trial.
Do you have somebody? Oh, here. I'm going to go over here too.
So people are human. Say you're getting ready to come to the hypoparathyroidism conference and you have an incredibly messed up morning and you forget to take the drug and you only inject once a week. How are you going to know? How do you keep track of that? Or when will you figure out that you didn't take it?
If you didn't, so during the clinical trial, it's easy because we know it will be done not by the patient, but it will be done by the healthcare provider. So that's why we know that the drug will be administered. Also, we have some PK. Remember that we are measuring some PK, so we can control that the drug has been taken. Now, in real life, when a drug will be on the market, well, it's your benefit to take the drug. And so I hope that you will be taking the drug, even your VPAS or this one, because that's for you. That's for your benefit.
Yeah, people mess up, though. They forget.
Yeah, so that's another question, which is when they mess up, such as forgetting a dose, etc., we'll be providing direction what to do if you forget for 24 hours or if you forget for 4 days. For example, if you forget 24 hours, you can inject just when you realize. If it's after 3-4 days, it's better to wait for the next injection.
I have a question. I may be confused with your other endocrine drugs that you were talking about. The GLPs, is this considered a GLP? And people that can't do GLP drugs, can they do this?
Oh, no, sorry. So GLP, we have 2 GLP in development. One which is an antagonist for hypoglycemia post-bariatric surgery. That's a GLP antagonist. And we have a GLP agonist with a GIP, which is a combination. Remember this, we can target 2 receptors at the same time, which is for obesity.
Okay,
and not for this.
Okay.
Not for hypoparathyroidism.
Okay,
it's
thank you.
Completely different indications.
All right.
Is there a washout period for your study? In other words, if a patient is on Forteo now, is there the typical 30-day washout before they can participate?
Yes, we have 4 weeks a washout period.
Okay. And then will there be any sites in Canada?
For now, there is no site in Canada. In the future, for when we get to phase 3, we'll be exploring and extending the number of countries.
And this one is saying, what will the follow-up be for the patient if they participate in the clinical trial? I'm not sure what that means.
So usually, as I said, at the end of the double-blind period, they can enter the 2 years open label extension, but if they decline for any reason, so usually it's 30 days.
And then someone is asking, can you speak at all to the time commitment for your trial?
So,
like beyond the 26 weeks, like, because I do know for your trial there is a, in the beginning it's a little bit intensive. They have to, like you said, have to get the injections at the site.
yes, so at the beginning, there is a commitment first to be in the optimization period. So it's 4 weeks usually, can be extended a little bit, but let's say 4 weeks. Then you get into the 12-week trial. And then the 30-day if you don't want to get into the open label extension. And if you want to get into the open label extension, again, if you're happy with your improvement, quality of life improvement, you get, and it can be up to 2 years. And it's beneficial that's why we created this open label extension, because it's not only 3 months and good luck. It's really taking a partnership with the patient to get into this open label extension. And also from a regulatory standpoint, we need also long-term exposure with patients. So it's beneficial because we will provide the tolerability, safety, also information.
So for the two-year open label, do they have to still continue to come in once a week?
No, it will be more spaced. At the beginning, a little bit, but then it will be more spaced.
Gotcha. Yep. All right. We good? One more?
If you only have 40 people, why was hypoparathyroidism interesting to you? Is it something that you just figured out how to solve, or does somebody have it, or why did you want to do
Now,
it?
I think 48 subjects have to enter into the study.
No, I mean, you're a small company. You said 40 people. What got you interested?
If you're interested, we are expanding. We are expanding. we are a biotech. We are a biotech, so we do not want to. We prefer to. When you're a biotech, what you want to do is to develop your product as much as possible and to invest into the patient and into the drug and into the clinical trial. So that's, and then at some point, because we have multiple programs, that's when you start growing. And that we are, well, at this stage, we are growing the company because we have more than one program. We have 3 programs.
Mm-hmm.
Thank you.
All right. Thank you so
Thank you
much
very
for your
much.
great presentation.